So a lot of how we classify viruses is determined by the structure of their outside capsules and the structure of their DNA or RNA inside. What we found is that a lot of viruses that infect animals, so things like influenza, coronavirus, tend to be RNA viruses, and they tend to have these viral envelopes. You can see over here how the envelope is acquired from the host. So the envelope is used to help kind of merge with the host and put the virus into the animal cell, where it's going to go through the lytic and the lysogenic cycles. During its reproductive cycle, the code RNA for the virus starts to produce these proteins that it's going to need on its envelope. And the host cell puts those proteins on its own membrane. And then when the virus bursts out of the cell, it's going to take that piece of membrane with it. And so that's how it gets this new envelope. It's not from its own body, it's taking it from its host's cell membrane. This table just shows us kind of how we classify viruses based on their nucleic acid. I don't expect you to memorize these classifications. I just want to show you some viruses that you're probably familiar with. So some double-stranded DNA virus would be some things like warts, cervical cancer, so human papillomavirus, herpes, smallpox, cowpox. We've got some double-stranded RNA, would be things like reoviruses, so that's going to cause stomach issues like rotavirus. And then a lot of the viruses that infect humans specifically are RNA viruses. So this group up here, we have single-stranded RNA, where the RNA is a messenger RNA. So you can see here's coronavirus. So that causes SARS and MERS. We should add to that COVID-19. The Zika virus, which was common during the Olympics, the last Olympics. Rubella, encephalitis. And then down here we still have single-stranded RNA, which has to be copied over onto another RNA molecule. So things like influenza, Ebola, measles, mumps, rabies. And then this last group down here is called a retrovirus. And it's a single-stranded RNA that has to be turned into DNA. So in this case, the RNA has to be kind of retrofitted back into DNA. And the example that we have that's really common is HIV, human immunodeficiency virus, in humans. So a retrovirus uses a specific enzyme called reverse transcriptase. And reverse transcriptase does the opposite of transcription. So transcription was when we took DNA and turned it into messenger RNA. Reverse transcriptase is going to take RNA and have to turn it back into a DNA. And that's what makes something a retrovirus, because it has to use reverse transcriptase. So that's HIV is a common human retrovirus, and this is going to require the body to turn the RNA back into DNA before it can build all of the proteins and new RNA molecules for the virus to go through its replication cycle. In humans, it's going to encode some of that DNA into the human DNA code, and we call this a provirus. And this isn't quite like a prophage. A prophage will eventually be removed from the host DNA, but the provirus does not. The provirus stays in the host cell as a permanent kind of member. And then that provirus DNA can eventually be transcribed into the particles, the proteins, and the new genome for the virus, and then released from the cell through the lytic cycle. So you can see how natural selection would work well with viruses. They don't fit our definitions of a living organism, but they definitely can evolve. And we can see how a virus that has genetics that code for proteins that allow it to infect cells is going to be able to pass its genetics on more easily than a virus that cannot infect cells. And so natural selection is going to select for viruses that are able to get inside of cells and kill them. We don't think viruses were a stepping stone to life, so that's been a hypothesis before, but it wouldn't make sense for life to evolve from a virus, because a virus needs a host already alive to be able to reproduce on its own. So viruses would not have been able to kind of populate the earth without hosts that already existed. So we think that viruses evolved after the first living cells, but probably from maybe damaged parts of cells, like little pieces of plasmids, which are pieces of DNA, or transposons, which are pieces of DNA that can move around inside of a cell. And so these might have all been kind of early movable genetic components. And these early movable genetic components may have led to the first viruses, which are in essence a mobile genetic component inside of some protein.